Assalamu alaikum. I am Dr. Muhammad Ali Rabbani, and today we are going to discuss the general anatomy of nervous system. It is the last lecture in the series of lectures on general anatomy. You can access the complete playlist by clicking on the link on the top left corner of your screen. Let's start with our topic. By the end of this session, you should be able to describe general organization of the nervous system. In this, we will discuss the organization of the nervous system, including its anatomical versus functional classification, parts, and the different parts, the different cells that are present, and basic functions of each of them. We'll describe the parts and formation of a typical spinal nerves, how they're formed, what they're made up of, what are the function of each of the component. Enumerate and describe the functional components of peripheral nerves. We will discuss separately for the cranial and the spinal nerves and give an overview of the autonomic nervous system. Let us start with the first learning objective. Overall, the nervous system, it is the network of nerve fibers and cells which transmit impulses between different parts of the body. When talking about these cells, the fibers are basically the extensions of the cell. Here we are talking about nerve fibers. These are basically axons and dendrites of the cells. And the nervous system can be defined in a different way also, like a computer. It is the network of nerve cells and fibers that takes the information and they process it and gives out the orders, give out the output to control the body and maintains the connection between each part. So that's about it. How it is made up, what is the general organization of the nervous system. When we talk about organization, we divide them on the basis of the anatomy. On the anatomical basis, it is divided into two parts: central nervous system and peripheral nervous system. It is better shown in the picture here. Let me show you. The central nervous system is the one that is present in the center. As the name indicates, it consists of two parts, that is the brain and the spinal cord. It is at the center of the whole network. All the incoming and outgoing impulses are connected to these parts. Then there is peripheral nervous system, the one that are all the nerves that are going and being distributed to different parts of the body. These constitute the peripheral nerves. They consist of cranial nerves. These are the nerves that originate from the brain, and the spinal nerves. These are the nerves that originate from the spinal cord. They connect the distal most away parts of the body to the central nervous system. Functionally, Functionally, they can be divided into the following two big categories: somatic nervous system and autonomic nervous system. Autonomic nervous system is also known as visceral nervous system. This is the somatic nervous system. Somatic means it's related to the wall of the body. It's somatic means body, it's related to the wall. In other words, it's the one that is under your conscious control. It can be further divided into two parts. Both of them can be further divided into two parts each. One is the outgoing signal, that is the motor component, also known as efferent component. The other one is sensory component, also known as afferent component. So these are the two divisions. The same will be here: motor or efferent, sensory or afferent. If we break them up, in the these are under your conscious control, this somatic. These are under the voluntary control or conscious perception and conscious control. These are involuntary control or in nature. Let me tell you by example. So for the somatic nervous system, the outgoing efferent motor fibers will be the one that you have a total control over. Like I'm talking muscles of my respiration, my lips, tongues, my hands. These are in the efferent division of somatic nervous system. These are in control. So it will include almost all of the skeletal muscles of the body. This sensory afferent nervous system, this component of front component of the somatic nervous system, it is basically the sensations, mostly the one that we are receiving on our skin and some of our special senses. I will break them down. These are the somatic sensations. I will explain it a little when I compare it with this afferent component of visceral system. Efferent motor component of the autonomic system, the visceral system, these are the outgoing signals that are not that much in control of our conscious mind. For example, your small intestine is continuously doing peristalsis to move the food. Your heart is beating at a special rate that changes its rate depending upon the different body needs and conditions. You cannot just think and change those to your need, change them to your will, like you can move your muscles and your other functions. So they are involuntary, they are automatically working, and they are mostly related to the viscera of your body. So these are the motor efferent component of the autonomic nervous system. Similarly, the sensations coming from the viscera back to the nervous system, those a lot of them are not reaching your conscious mind. For example, you don't know when your blood pressure rises a little and your heart responds by changing the force of contraction. We don't perceive it consciously until and unless they go beyond a certain limit. Similarly, when the food reaches from stomach to the small intestine and the signal goes to the pancreas to release its secretions, we don't perceive the movement of food from one component of the intestinal system to the next one. So these are the autonomic sensations, and the examples are numerous. These will include the chemoreceptor sensing the composition in the blood, the baroreceptor sensing the pressures in the blood, and on at different components. However, some forms of sensations are perceived by our conscious mind, like when you get too full, you get acid reflux, the burning sensations that we feel in the chest. Like flatulence, if we have a gas in the body, we get a pain and the distension of the intestine occurs that leads to the pain in the body. Now that is what I want to compare with the somatic part of the efferent division and the visceral part of the afferent divisions. I'm sorry for that. Sensory component of the somatic system, or compare with the sensory component of the autonomic system. In the sensory component of the somatic system, the details are very high. For example, even if at the back of your hand an insect is crawling, you don't look, you just do like this. You can sharply localize it. You can feel just by the sensation what type of input is being given, like an insect is running, or even the size of an insect you will feel. You can move your finger across the table and give a judgment about the texture. But when talking about the visceral sensation, when you have heartburn, you can't give many comments regarding the nature of the burn apart from it's more or less. You can't totally localize it. I just feeling some pain right here. I'm just feeling some pain in the abdominal region. Compare it with this part. When I have a water droplet on my earlobe, I just go and clean that. So this is the how they are different from one another. Some authors, they divide them in the other direction. Rather than dividing them into somatic or autonomic, they divide them into afferent and efferent, and then they divide them into somatic and autonomic. And you can divide these into somatic and autonomic. So it ultimately they give us the same combination of four categories. So this is about the division of the autonomic nervous system. Let us talk about the cells which are present in this system. This is the main cell. This is the one that is creating all the fuss. This is the one that is the functional unit of the nervous system. It is the one that is conducting the nerve impulses, carrying the information from one part to another part. All the other cells that are present in the central or peripheral nervous system that give support to the neuron in any way, those will be in the category of supporting cells. Those cells themselves are non-excitable, are non-conducting. They do not generate impulses, but they help impulse generating cells by protecting them, nourishing them, maintaining their environment, and helping them in many other ways. Let's talk about the neurons. Neurons are the excitable cells. These are the cells that conduct impulses. Neurons are identified on the basis of two basic characteristics. They have excitability and conductivity. These cells, let's talk about conductivity first. These cells have, all the cells in the body have a change of potential, electric potential across the membrane. These cells have an ability to rapidly change that impulse, and that change in impulse as a wave can be propagated across the surface. So this property is called conductivity. A change of electric potential, it travels, it propagates along the length of the cell all the way to the periphery. This is called conductivity. So this is one function, but the wave of electric impulse, it has to be initially generated. The wave has to be pushed to be brought to a certain threshold, after which it becomes self-propagating. So this ability to excite the cell or change the action potential to reach that threshold, this is called excitability. This excitability can be brought upon by the impulses from the other cells as they move on and reach this cell by a synapses, or they can be by any external stimulus. For example, in the case of receptors, the rods and cone receptors, they generate an impulse in response to the visual stimulus. Same goes for mechanical receptors and the auditory receptors. They generate impulses. They get excited when they get their respective factor trigger to move them. Talking about the anatomical structure of the cells, each consists of two parts. This is the main body. This is the body of the cell. Like all the other cells of the body, it consists of all the organelles and nucleus in the center, cytoarchitecture, and all the organelles that makes up the cell. This is the soma, perikaryon, or the body. These processes that are extending from the cell body, these processes are known as neurites. The common word for all the processes is neurites. Now, depending upon the direction of the impulses, these can be divided into dendrites. Dendrites are the processes that brings the signal towards the cell body. They brings the signal towards the cell body. Usually cell have a lot of dendrites, may have single also, but usually have a lot of dendrites, and they have very branching expanding pattern. They make connections with a lot of cells or lot of structures in the surrounding areas, and get their effect is summed up in the cell body. These are the dendrites. And the one large process that takes the impulse away from the cell body, that is known as axon. That is known as axon. Axon, usually one axon is exiting from the body. Yes, as the axon moves out, it may divide and give one or collateral branches. These are like this is getting sideways. This is called collateral branch, and ultimately it goes and it synapses with the next neuron or the effector organs. For example, in most of the cases the axon makes a synapse with the muscle. There we do not call it a synapse, we call it a neuromuscular junction. So this is the terminal part. The terminal part is divided usually into a number of branches, so that a large surface area, a large contact region can be created. So these terminal branches are called terminal arborizations. So these terminal branches, these are called terminal arborizations. So this is the basic, very simplistic view of the cells. The cells, as you can see, the axons, these axons are being covered with this number of cells. These are covering the axons. These are basically the insulating cell, like the insulation uh, we give to the electrical wires. Nature has given insulation to these conducting neurites also. These insulating cells are called Schwann cells. These are called Schwann cells. Schwann cells, a number of Schwann cells, this is one, this is the next one, this is the next one. The number of Schwann cells in close cover a single axon, and when one Schwann cell ends and the next one starts in between them, this area is called node. This is node, where to be more specific, it is called node of Renvier. These are the nodes of Renvier. So these are the neurons. On the basis of their branching pattern, anatomically speaking, they can be, morphologically speaking, they can be divided into the following types. If the cell has only one process, one neurite, it is called unipolar. If the cell has two neurites, two processes, it is called bipolar. And if it have more than those are called multipolar neurons. Let me show you by this picture. This is the cell. This is a typical multipolar neuron. It has a large number of dendrites attached to the body, but even if the cell is multipolar, it usually have only one axon. Yes, the axon, as it moves around, it may branch to give terminal arborizations or the collateral branches. This is the format that is followed by a lot of neurons in the special senses. It has one dendrite entering the body and one axon entering the body, exiting the body. And dendrites, if we follow it, trace it back away from the cell body, it may show the branchings beforehand, but only one dendrite is being connected to the cell body. So this is called bipolar. This is called unipolar or sometimes pseudo-unipolar because this cell has only one connection, one neurite. It is further divided into two major branches. The one that is towards the central nervous system is the central process. That is towards the periphery of the body is peripheral process. They're usually the sensory nerves, and they carry the signal from the peripheral to the central. So one process that is from the peripheral process is acting as a dendrite, and the other process is acting at an axon, taking the signal. That is why, because the signal is being transmitted in both directions, in the dendrites away from the cell and towards the cell. So sometimes this is a part, rather than calling them unipolar neuron, they are called pseudo unipolar neuron. And then there are cells that are present in the central nervous system. They have relatively complex functions. They do not have any axon, rather they have a lot of dendrites, and then they basically act by changing the action potential of the neighboring cells. They do not have any axon, so that is why they are called anaxonic. Now let us talk about the types of neuron on the basis of their function. This is the basic division that we just discussed with the divisions of the anatomical, the division of the nervous system, like this basically in the direction of the nerve impulse being moved. For all the nerve fibers that are moving the impulse away from the central nervous system, those are called motor or efferent nerves. For example, the cell body of you move your biceps as you fold your arm. The cell body is present in the spinal cord and it carries the impulse away from the spinal cord all the way from the nerve until it reaches the neuromuscular junction supplying the muscle. So this is the efferent motor neuron. The sensory neuron is the one, the afferent neuron is the one that is sensory in nature. It brings the signal towards the central nervous system. This is a very good example. You touched a hot object, the signal is brought to the central nervous system by afferent nerve. By afferent nerves, as you can see, it's pseudo unipolar usually in the spinal nerve, and it enters the central nervous system. This is the afferent nerve. In the with so these afferent and efferent nerves are present outside the at least their process there it's present outside the central nervous system, and if it is carrying impulse away from the center it's efferent, it's carrying towards the central nervous system it's afferent. That leaves us with the cell bodies, the neurons that are present within the central nervous system. So these neurons, these are called interneurons because through a various connection in the central nervous system they are connecting ultimately. If it's a single neuron, it's a single neuron in case of very simple reflex pathways, and there are lots and lots of neurons in case of like process like conscious thinking. But ultimately all these neurons are responding to the afferent neurons and giving signals to the efferent neurons. So these are the interneurons that are present between these two, and they are present within they are present within the central nervous system. They are present within the central nervous system. So these are the different neuron types. Now we talked about neurons a little. Let us talk about the cells that are supporting them. These cells are different in the peripheral nervous system and the central nervous system. In the peripheral nervous system, there are two neurons. I will shortly show you the pictures. The ones that are forming the insulations around the axons, these are called Schwann cells. They form sheath, they form insulations, and the sheath that they form is called myelin sheath. And there are neuron cell bodies that are present outside the central nervous system in the sensory ganglions, in the dorsal root ganglions, these are sensory ganglions, and in the autonomic ganglions like sympathetic chain, the ganglions present there. Those cells are surrounded and supported by satellite cells. In the central nervous system, there are four types of cells: oligodendrocytes, astrocytes, microglial cells, and ependymal cells. Oligodendrocytes are the ones that are counterpart basically of the Schwann cells in central nervous system. Like Schwann cells, they were making insulation around the axons in the peripheral nervous system. Oligodendrocytes make sheets, makes insulation in the central nervous system. Astrocytes are star-shaped cells. These are the ones that make blood-brain barrier. They maintain the environment, they give support, and they form the scar tissue. Microglial cells are basically macrophages. These are the scavenger cells that eat up anything bad, any pathogen, any debris or extra material that is. So they clean up it and they remove it. And ependymal cells are the one that lines the CSF containing cavities, including the ventricles of the brain and central canal of spinal cord. So these are the two cells of the peripheral nervous system. These are shown in these pictures. These are pictures from the Gencoira's histology. This is a peripheral nerve cell body and the axon that is present in the cell. So these cells, this is one cell, this is the next one, this is the next one. These are the Schwann cells, and as you can see, each Schwann cell it's rolled around the axon multiple times to create a layered insulation. So these are myelin sheaths. So they form the sheath, and the cells that are laminated by these cells, these are called myelinated axons, myelinated cells. And one Schwann cell is very, in the peripheral nervous system, the Schwann cell is very faithful. One Schwann cell only surrounds one axon. It does not go between different cells. Yes, the cells that we classify as unmyelinated, they still are near the Schwann cell, but as you can see from this picture, they do not have that much laminated or proper insulation. And here the other axons may be partially or embedded in the Schwann cells, but they do not have that lamination. And here the one Schwann cell per axon does not apply also. But so these are called unmyelinated cells. The gap between the unmyelinated cells, two cells, that is called node, and the length segment of the axon between two nodes, that is the area beneath one Schwann cell, that is called internodal area. Then there is a cell that are shown in this pink purple color. In the peripheral nervous system outside the spinal cord, the cell bodies which are present, these are surrounded by these cell. These are called satellite cell, which surround these cells and maintain its microenvironment and help its survival. So these are satellite cells. Now this is a picture again from the Gencoira's histology showing the cells in the central nervous system. Shown in the white blue color, light blue color, these are the oligodendrocytes. These are basically the insulating cells in the myelinating cells in the central nervous system. And you can see the difference from the Schwann cells, that they are not faithful like Schwann cells, and they have one oligodendrocyte is connecting to a large number of axons. But and they have the same function, the same nodes, the same internodal area and everything. Shown in the green color here are the astrocytes. They are given their name because of their star-shaped like appearance, because they have a large number of processes, and these maintain the microenvironment of their surroundings, maintaining especially the calcium, potassium levels in their surroundings, giving support to the cells. And very important function is they surround the blood vessels that are inside the central nervous system, and they make junctions to give make very strong blood brain barrier. So they have a very strong function there, and they have an additional function of lying down the scar tissue, because the neurons, they do not multiply, they are formed, the number is finalized after the brain is matured. So these astrocytes, these are the ones that lay down the connective tissue, they lay down the extra cell fibers when a defect is to be mended, when a gap is to be filled. Like a person had a surgery of a tumor, there is a gap created, that gap is filled by these astrocytes. The next one are microglial cells. These are the microglial cells. These are basically not a component of, not a derivative of the nervous system. They are migrated from the blood into the central nervous system. They are the macrophages that have migrated here. They do all the functions of the macrophages, including pathogens, phagocytosis, antigen presentation, removing the debris and the waste materials. They are doing all those functions. They have some role in the blood-brain barrier also. That leaves us with these cells. These are the ependymal cells. The central nervous system contains a fluid running inside them. There are cavities in the brain containing CSF. These cavities are called ventricles, and the cavity in the center of the spinal cord, that is called central canal. So these cavities, they contain the fluid, cerebrospinal fluid, and those cavities are lined by special cells that are called ependymal cells. And these cells have a very important function in the generation of those fluid, in beating and movement of those fluid, and sampling and absorption from that fluid uh, in various capacities. So this is all about the cells in the central and peripheral nervous system. Of course, as you go into the histology and nervous system module, you will go into much deeper details. These are the few terminologies that you must familiarize yourself with as you move along, because these will be encountered frequently. The difference between the nerve and a tract. The nerve. We saw that the cell body may have a large neurites, the axons that are running along. So the cell bodies are not just scattered among the nerves. The cell bodies are very well placed either inside the central nervous system or outside the central nervous system in form of groups like called ganglia. So when a nerve that is traveling, it contains only the neurites of the on the or the nerve fibers of different nerve cells. A mixed nerve will contain the neurites, the axons of the motor nerve, they will contain dendrites going to the cell body of the sensory nerve. This bundle of neurites containing both the afferent and efferent fibers, that bundle is called nerve, and that nerve will be present in the peripheral nervous system only. In the central nervous system, different cell bodies are connected to each other. So one cell body have to have a neurite, an axon that has to travel some distance to get attached to the next neuron. Sometimes these are very large distances. For example, the sensory signal that is entering from the joints, carrying the sensation of proprioception, the sensation of position, it enters the central nervous system and travel all the way till here as it enters the cranial cavity, and then it makes the first connection with the next cell body. So this bundle of nerve fibers, the neurites that is running within the central nervous system, that is called tract. So there are different tracts you will study in the nervous system, that you will study about the dorsal column, medial lemniscus system, spinothalamic tract, and all the different tracts that are carrying the signal towards the brain and away from the brain. Similarly, ganglia or nucleus, these two words are differentiated from one another. The cluster of cell bodies that is present outside the central nervous system, that is present out in the peripheral nervous system, that is called ganglia. So this includes the sensory ganglions like dorsal root ganglion, or autonomic ganglions like the ganglions present in sympathetic chain, or the parasympathetic autonomic ganglion that are present in head and neck region. Nucleus is the collection of cell bodies, neuronal cell bodies, that is present within the central nervous system. For example, within the brain, there is a white matter, but there is accumulation of cells that are called basal nuclei. They are having a role in fine-tuning your motor activities. Junction of the white two neurons, the axon as makes connection with the dendrite of next cell, or axon makes connection with the cell body of the next cell. This is called synapse. These are neuronal junction. Gray matter and white matter, and this there is a spelling mistake here. This is called gray matter, not matter. Okay, gray matter, white matter. Let's ignore the spellings right now. I will show you in the pictures, upcoming pictures. In the central nervous system, the areas which have abundance of the cell bodies, those are darkly stained, or not stained. Those in a fresh section, they appear darker because they contain cell bodies embedded in neuroglia. Neuroglia means all the cell fibers. So they are appearing dark in nature, and where a lot of tracks are running, because they are myelinated and myelin sheath is ultimately white or pale in color. So that area carries a relatively lighter tone. So that area is called white matter. So this is an example of the synapse, like an axon, as it makes connection with the other next neuron. The axon makes connection with the dendrites. The terminal part of the axon, it is expanded, buton, and that contains a large number of vesicles and contains the transmitter substance. The two neurons are not technically touching each other. They have a very small gap between them known as synaptic cleft. So what happens is that when a signal electric impulse comes, it travels along the cell membrane, gets to the last point of the axon, and this impulse lead to this stored secretory vesicle storing the transmitter substance to empty their material outside into the synaptic cleft. These transmitters, they have their corresponding receptors in the next cell membrane. They have receptors. They get attached to the receptors, and the attachment leads to some conformational change that excites the next cell to produce an impulse that can then be propagated along the way. So this is how a synapse work. So you must keep in mind there is a presynaptic axon or neuron, there is a postsynaptic axon neuron, and the cleft between them. The presynaptic will have the secretory vesicle that are emptied in response to the incoming impulse. It traverses the synaptic cleft. It attaches to the receptor, which respond to the attachment by generating or sometimes downplaying an impulse in the next cell, and the next cell then moves or creates the impulse. So this is called synapses. Now let's talk about the central nervous system. It consists of brain and spinal cord, and the neurons, the excitatory or conducting cells, the functional cells that are embedded in the connective tissue. This connective tissue is given the name neuroglia. They are organized into gray and white matter. We just discussed what are those, and the arrangement of the gray and white matter is opposite in brain and spinal cord. In brain, the gray matter is outside, the white matter is inside. However, in spinal cord, the white matter is outside and the gray matter is in the center. This can be shown here. This is a section from the brain. The darkly stained area is present near the surface. It will have more nuclei. The lightly stained area containing most of the tracks, this is present in the inside. And here you can see the different nuclei that are in collection of cell bodies embedded in the white matter. In the spinal cord, however, this arrangement is opposite. The tracks are on the outside, white matter, and the cells are on the inside, gray matter. Talking about the peripheral nervous system, it consists of the distributing nerves. The nerves consist of, as you remember, the neurites or the cell processes, and ganglia. These are the cell bodies that are present outside. Anatomically, depending upon their origin, they are classified into two main groups. Cranial nerves, these are the nerves that emerge directly from the brain. There are 12 pairs. And the I have uh, I must have missed it. The peripheral nerves, rather than using the word peripheral, the correct word is spinal nerves. Spinal nerves. These are the nerves that are exiting from the spinal cord. They're exiting from the spinal cord, and there are 31 pairs that exits from the spinal cord. So this is the cranial nerves. Cranial nerves are named by according to the number. They're 12, so they're named 1 to 12, and they are written as their Roman numerals. And it you must as you go along, you will be exposed to them more and more as you will advance in your courses, and you have to remember their names also. The olfactory nerve, optic nerve, oculomotor, trochlear, trigeminal, abducent, facial, vestibulocochlear, glossopharyngeal, vagus, accessory, and hypoglossal nerves. What are the function of these nerves? This is beyond the scope of this lecture, but you must, for now, as a beginner, you should try to remember the names of the nerves. Even if you don't, just remember the fact that there are 12 pair of nerves, and all of them are emerging from the brain. And as you can see, the labeled this part, all of these, the cranial nerves, they are exiting from the brain substance or the brain stem. We will differentiate between brain and brain stems as we move on to the nervous system module. Functional classification. This is what we talked about earlier. Functional classification can be done on the somatic and visceral, and on the both of these can be further divided into afferent and efferent. So these are the somatic. Afferent are the sensations that you feel on your skin, and you have very detailed information about these. You are consciously aware of those. Somatic. Efferent, the outgoing signals you have your conscious control over. Visceral nervous system, this is the afferent divisions, the sensations from the viscera. You might not always receive them consciously, or even if you do, you don't get them in that much detail. Visceral afferent, the involuntary, the autonomic nervous system that is controlling your different viscera. Now let's talk about the spinal nerves. There are 31 pairs of the spinal nerves that are exiting the spinal cord, and they passes in the foramen created between the two vertebra, that is intervertebral foramen in the vertebral column. And you must remember these figures. In the neck region, in the cervical region, there are 8 spinal nerves. In the thorax region, there are 12. In the abdomen lumbar region, there are 5. In the sacral region, there are 5. And in the tip, the tailbone, that is one coccygeal bone. One coccygeal pair. Sorry. Here is an interesting thing. The vertebra, the vertebral column, the bony skeleton, it goes all the way down to the tailbone. That is the tip. You can feel it on your back. However, the spinal cord does not go all the way to the lower border to the coccyx bone. It ends at the lower border of first lumbar vertebra. This is the figure that you must remember. They end at the first lumbar vertebra. So what interesting consequence is that to accommodate the difference in size, the let's say the sacral nerve, the coccygeal nerve will exit between the coccygeal vertebra. However, the spinal cord is all the way and up, ending at the L1. So after exiting the spinal cord, the nerves will travel down and then exiting the spinal cord. Let me show you in the picture here. The spinal cord is ending right here at the level of L1. It is ending right here. But you have to remember that the spinal nerve is exiting the bony vertebral column at their designated place. Like first sacral nerve is exiting between first and second sacral bone. The second sacral nerve is exiting between second and third sacral bone, and so on all the way till coccyx. However, the spinal cord is ending all the way up here. So after exiting the spinal cord, they have to travel down on their own till they reach their place of exit. So as we move above from upper end of the spinal cord to lower end of the spinal cord, you will see the spinal nerve lengthening and traveling within the vertebral column before exiting. Like in the topmost region, they're almost horizontally exiting the vertebral column at the level of exiting the spinal nerve. As you go down, you can see the increase in slant, and as you go beyond the lower end, you can go a tuft of nerve fibers. They are exiting, and at each vertebral body, at each bone, between each pair of bones, one nerve pair exit, and this tapers down as the last nerve exits at the level of coccyx bone. So this tuft of nerve that is going down, that is called cauda equina. Cauda equina means, literally, cauda means tail, equina means horse. So it's like a horse tail. These nerve fibers that are resembling the horse tail, and they move from the termination of the cord, known as termination of the cord. Also, it is known as conus medullaris. And the conus medullaris, that is the terminal portion of the cord, and it is moved down. This is called cauda equina. So this is about the nerves. So here is a picture, alternate pictures that are shown to make you understand them easily. The lower end conus medullaris, the tuft of fibers going cauda equina. Structure of the peripheral nerve. How the peripheral nerve form? Overall, if you cut a section, it is composed of large number of bundles of the nerves, fascicles of the nerves, which are held together by connective tissue. If you have listened to the lectures on the arrangement of muscle, the general anatomy of muscle, they were organized in multiple steps. The single muscle cell was surrounded by endomysium, then a bundle of fascicle was surrounded by perimysium, and the whole muscle was surrounded by epimysium. The same formula applies to the nerve. Each single axon plus its Schwann cell, it is surrounded by endoneurium. A bundle of axons are surrounded by perineuron. That bundle is also known as fascicle, and the whole nerve is surrounded by epineurium. So it is shown in this picture here. This one axon surrounded by Schwann cell, this setup is surrounded by a connective tissue layer that is called endoneurium. Multiple myelinated axons, multiple myelinated fibers surrounded by endoneurium, they bundle together and they have their own covering of the bundle. That covering of the bundle is called perineurium. Depending upon the thickness or overall size of the nerve, multiple bundles get together, and the bundle of bundles, this is surrounded by epineurium. This is surrounded by epineurium. How the spinal nerve is formed anatomically speaking? Each spinal nerve, it starts from the spinal cord by two roots: anterior root, ventral root, that that contains the outcoming, the efferent, the motor fibers. The motor fibers, their cell body is present embedded within the spinal cord. The posterior root, it consists of the incoming fibers, the sensory fibers that are entering into the spinal cord, and their cell body is outside in the dorsal root. As they exit the vertebral canal, as they move from the intervertebral foramen, the ventral and dorsal roots, they join one another to form a mixed nerve. Now let us see them in a picture. This is a typical section of the spinal cord. Outside there is a white column or white matter. Inside there is H-shaped, butterfly shaped gray matter. From the gray matter, on the ventral aspect, on the anterior aspect, these are cell bodies that give rise to the axons that have to take their signals to the effector organs outside. So in this case, these are the, let us suppose these are the fibers going to the biceps muscle. These are the axons that are carrying the signal outwards. These are the motor roots. These are the motor roots. Similarly, the sensory signal is coming back from the body. They have their pseudo unipolar in nature. Their cell bodies are located outside the nervous system. central nervous system and they exit the spinal cord here, and their direction of impulse is towards the spinal cord. So this is the sensory route. As they join here, now this is called the as they join here, now this becomes a mixed spinal nerve. This is called a mixed spinal nerve. The mixed spinal nerve contains the incoming fibers and it contains the outgoing or efferent fibers. So this is the how a spinal nerve is formed, and this nerve is this union point is at the level where the nerve is exiting the vertebral column through the intervertebral foramina. Now let's see what happens to it further. After emerging, the mixed nerve immediately divides into anterior ramus. This is the branch that supplies the anterior and lateral walls of the body, including the limbs. And it is having connections to the sympathetic nervous system, and it is forming the different plexus. The posterior ramus is the branches that are going to the back and supplying the skins and muscles of those areas. This is posterior ramus. The anterior ramus at the roots of the limbs, they mix and match and they form very complex nerves plexi. For example, in the neck region there is cervical plexus. In the neck and the upper thorax region there is a brachial plexus. This brachial plexus supplies the upper limb. In the abdominal region, in the lumbar region there is lumbar plexus. In the pelvic sacral region there is sacral plexus, and the lumbar and sacral plexus, it supplies the lower limb. So let us see them one by one. Now this is the same formation shown in a different picture. This is a picture from Snell's clinical anatomy by regions. From the ventral root, there are cell bodies that are present inside. The motor root emerges containing the signals going away from the cell body, shown by the red color here. And on the back, there is incoming fibers whose cell bodies are present outside the unipolar one, the dorsal root ganglion. These roots that form the posterior root, they are carrying the incoming fibers shown by the blue. At this particular point, they join one another and they form a mixed spinal nerve. They form a mixed spinal nerve because now it contains both the sensory and motor components. Immediately after doing that, it is divided into two parts. This one I have labeled 1. This is the anterior ramus that will go anteriorly, supply the limbs, the lateral and the anterior aspect. And the one that is going to the back, so that will be the dorsal ramus that will supply to the muscles and the skin of the back. So this is the dorsal ramus. And in addition to that, the anterior ramus, it makes these connections. We will go into the details shortly in this very lecture as we move on to the autonomic nervous system. And as I mentioned previously, the ventral ramus, the anterior rami, they join to form different plexi. Immediately, you are good enough if you know the names of the plexus, and very good if you know the root values of the plexus, which nerves join to form the different plexus. Right now, you don't need to remember all the branches of the nerves, but as you move along into the regional anatomy, by the time in the next 2 years you're supposed to complete your course of anatomy, by that time you must know all of these plexes in detail and the nerves that are emerging through them. Now there is a word that is dermatome. Through extensive experimentation and work, scientists have discovered that each spinal nerve, it supplies a particular segment of the skin that is different from the segment of the next one. So all the area of the skin that is supplied by one particular spinal nerve, one particular spinal segment, that is called dermatome. Like here, T2, the nerve exiting from the T2 second thoracic segment of the spinal cord, second thoracic spinal nerve, this is the area of its supply. On the front, on the back, this will be the area of its supply. Immediately they might not look very useful to you, but as you move along, they become very helpful when we are studying the diseases, when we are studying the injuries of the spinal cord. Like a patient comes to you with a broken back and he or she has damaged the spinal cord at a particular level. There are, of course, MRIs and radiological tests that will help you find out where exactly the damage occurred. But in the clinical examination, you might start from one dermatome and go to move on to see which dermatomes the person is feeling sensations in. And let's say he's feeling the sensation in T1 dermatome but is not feeling any sensation is T2 dermatome. Then we will find out that the spinal cord was damaged at the level where T2 nerve was exiting it. So it helps to give a very good idea of the different injuries, and even in the normal functions, it very much helps us to understand the referred pain. Why, like for example, the heart, it's receiving signals from T1 to T4 nerves. So during the heart attack, the person usually feels pain in these dermatomes, T1 to T4 dermatome, majorly in the left side. But you might have seen in the movies, or sometimes you might have mm, seen some people when they're having heart attack, they feel the contraction in their chest and the extension to their limbs. So these dermatome explain that fact. So there are multiple maps available of the dermatomes that help you understand the theoretical mapping that helps you get the idea of where each segmental nerve supplies. The signal from the skin, the signal from the body, the external environment, it reaches the inside via receptors. So basically, receptors are the organs, these are the structures that receive the environmental stimulus and generate a nerve impulse in its response. For the purpose of general anatomy, we will be discussing we will just touching the surface about when talking about their types. Exterior receptors in general will be the term that will be applied to those receptors that gets impulses from the external of the body, external stimulus. And in by convention, this term is mostly used for those receptors which are associated with touch, like you feel the texture, you feel the vibration, you feel the pinching, the pain, you feel the temperature on the skin. So these are carried by exterior receptors. There are a number of receptors that are included in this. We will discuss as we move along with our subject. Teloreceptors, like television, distance vision, like teloreceptors are the receptors that gets the impulse, the external stimuli at some distance away from the body. You might have already guessed them. Like the vision, you pick up the signal from the distance. Like smell, you pick up the signal from a distance. Like hearing, you pick up a signal from a distance. Interoceptors, visceroceptors are the receptors that are present inside the body. Like the pressure receptors that detect the blood pressure in your body. The chemoreceptors that detect the mm, uh, chemical composition. For example, just try to hold your breath. You will start panicking after about half a minute. So this panic is these bells are being rung by the chemoreceptors that are present inside the body that are raising the alarm that the oxygen level is down and carbon dioxide level is very high. And there are a number of receptors picking up the hormones, picking up different uh, ions levels and oxygen nutrient levels inside. And proprioceptors are special type of interoceptors that responds to the stimulus related to the position and movement. For example, if I close my eyes and I ask my friend to change the position of my hand, and then I try to describe, then I can very well explain that my fist is closed, all my fingers are curled. So I have a very good sense of the position of all the joints of my body. I can tell you that I'm sitting without even looking down. My arms are by my side, my elbows are flexed. This I can tell without telling you. So these internal stimuli, the length, the tension in the body, this position, so these are proprioceptors. These are muscle spindles and Golgi tendon apparatus, etc. The effectors, the major one that we see them very visibly, they are the skeletal muscles that bring about the different movements in the body. So effectors are all the parts that carry out the functions in response to the impulse from the nervous system. Contraction of the cardiac muscle, contraction of the smooth muscle. So all the muscles are basically affected that are producing some activity in their capacity. The other type of effectors are basically secretions, the glands that are producing secretions. So they are producing their response, they are producing their effect by producing their secretions. Now let us talk about the functional components of the nerve. Functionally, typical spinal nerve has four components. You might have already guessed their function by their names. We talked about them when talking about the divisions of the peripheral nervous system. Only the word journal has been added. So the fibers that are of the somatic nervous system and carrying signals away from the cell body are somatic, general somatic efferent. Like these will be the fibers that will deliver their fibers to the muscle cell that are under voluntary control. So the signal carrying impulse to my muscles so that I can move them on my will, those are general somatic efferent fibers. General visceral efferent. This will be the autonomic nervous system. This will be the involuntary system that gives impulses to my smooth muscles around my viscera, that will give supply to the cardiac muscle in my heart, that will give supply to the different secretory glands in the endocrine and exocrine domains of their function. General somatic afferent. These will be the sensory nerves of the somatic system. This will include the fibers getting their sensations from the exteroceptors and interoceptors that we get a very detailed information on. Like we talked about the ant crawling on the skin, the information about proprioception, the information about um, proprioception, the information about vibration, pressure, the information about pinprick. So these sensations that gets registered very detailed manner from the somatic system to the back to the central nervous system. These are general somatic afferent and general visceral afferent. These are the sensations from the viscera that are ultimately reaching the central nervous system through spinal nerves. Again, we talked about the bladder distension information, the stomach distension filling information, the blood pressure information, the blood chemical composition information. Those are all general visceral afferent fibers that are carrying those information. The 12 cranial nerves, they contain 7 rather than 4 components. They include these 4 components: general visceral, general somatic efferent, general somatic afferent, general visceral afferent. They have the same one. Let's talk about them by the examples of cranial nerves. For example, the muscles moving your eyeball will be the muscles that you have your under conscious control. They will be in general somatic efferent. Taste sensation, not general taste sensation. Let's say the sensation from the back of the pharynx, the sensation of the carotid body and carotid sinus, carrying the information around the blood pressure and composition. These will be general visceral efferent. Afferent, sorry. I just gave the example of general visceral afferent. General visceral efferent will be, let's say, the parasympathetic outflow that gives the salivation signal, that gives the tears, the lacrimation signal. Those will be general visceral efferent. General somatic afferent will be the sensations, let's say, hot and cold that I receive on my face, on my forehead. Those will be general somatic afferent. Now the other ones, that are unique to the cranial nerves are these three. The names are the same, but the special is added. So there is a special apart from, there is no counterpart of general somatic efferent, but there is a special counterpart of each of the next three. General somatic afferent will have a counterpart, special somatic afferent. Special somatic afferent, these are the special senses. These both special afferent, special afferent, these are both special senses. General somatic afferent, these include the sense of sight and hearing. Sight and hearing. What you listen, what you hear. This is special somatic. So the sensation from eye, the sensation from ear. And these will include chemical sensation, or chemical sensation will include the sense of smell, the sense of taste. So these will be included in special visceral afferent. Then we are left with special visceral efferent. It will be a little difficult to understand the first time. As we move along in the subject of embryology and discuss the origin of different muscles, there are the muscles in the head and neck region that are originated from the pharyngeal arches. So those muscles, the supply of those muscles is called special visceral efferent muscles. Autonomic nervous system, that is the next major chunk of our lecture. This is basically, in talking in the language of the division and the types of fiber, that is basically general, you might guess it as we write along, visceral efferents. So these are the supplies, the involuntary supply that is going to the viscera of the body. The visceral part of the nervous system, it supply innervates the involuntary structure through both the central and peripheral nervous system. It includes like the heart, the cardiac muscle, the smooth muscle, the secretory organs. In general, the somatic system was one neuron linkage. Like from the central nervous system, the axon reached directly the effector organ. The autonomic nervous system, there are two neurons. Let me show you better with example. There is an impulse from CNS that first relays in a second neuron outside the CNS, but then that reaches the organ. So in case of somatic system, somatic efferents, the cell body is present in the central nervous system. The axon moves all the way uninterrupted and it reaches the effector organ. In this particular case, it is the muscle. However, the autonomic nervous system, it always contains two neurons. First neuron, the cell body keep a very clear indication of the body component. The cell body is present within the CNS. The second one, the cell body is present in the peripheral nervous system. So the first cell body is present in the central nervous system. It exits the central nervous system, goes all the way until it reaches the cell body of the neuron number 2. That is the second neuron. Between those cells, the connection is called synapse. There is a synapse between the two neurons, and after that the second cell body gives rise to another axon that go and reach the effector organ to produce its effect. So the first neuron is before the synapse, the second neuron is after the synapse. So the first neuron is called, can you guess it? Its name is written in the labeling also. It is called pre-synaptic. It is written above also. And because the cell body are present in the form of the cell body are present here in the form of ganglion, so those are called ganglions, so they have an alternate name of preganglionic. See, these are presynaptic and preganglionic, the first one, And the second one, it's very simple. The second one will be thus called post synaptic or post ganglionic. So these are the two neuron system: presynaptic, preganglionic, postsynaptic, postganglionic. This one, the pre one, is usually myelinated. This one is myelinated. So it is white, it is insulated, it is having high speed impulses. This is usually unmyelinated. These features that we have discussed about the autonomic nervous system, these are common to the both divisions, the sympathetic nervous system and the parasympathetic nervous system. Let me revise them. They will have two cell bodies. First cell body will be present inside CNS. The second cell body will be present outside CNS. The second cell body will be present in with other second cell bodies in the form of an aggregate called autonomic ganglion. The axon of the first cell body will be called presynaptic or preganglionic axon, and it will be myelinated. The axon of the second cell body will be mm, unmyelinated, and it will be called postsynaptic or postganglionic axon. So these two divisions of the autonomic nervous system are sympathetic nervous system and parasympathetic nervous system. I don't want you to memorize all of the functions immediately. Of course, as you move along, you will have to do it. But immediately, I just want you to recall what happens in a panic, fight, and flight state of the body. That are the functions of the sympathetic nervous system. What are the functions that the body do when it is resting and digesting? So those are the functions of parasympathetic nervous system. So in the sympathetic nervous system, the heart rate increase, the blood ratio increase, the peripheral vessels constrict, the blood vessel go to the muscles, you forget about going to the washroom, your sphincters are closed, your blood peripheral vessels constrict, the blood is supplied to the core. Those are all the functions of sympathetic nervous system. Your pupils dilate and your hair stand up and all of those, and the list is much larger than what I just told. And when you are relaxed, you just slow down, your heart rate gets normal, your heart rates drop down, blood pressure drops down, uh, intestine peristalsis starts occurring, you feel hunger, your sphincters begin loosening, you want to go to washroom now. Now these are the functions of the parasympathetic nervous system. You can pause the video here, and I will remove myself. You can pause the video here and just go through the whole list. So at different organs, what is the influence of sympathetic nervous system and what are the influence of the parasympathetic nervous system? I will not be going into the much detail. You can pause the video and go to the detail yourself. I will be more focusing on the anatomical component. Let's start there. Here, the outflow, I just told you that the first neuron, the presynaptic or preganglionic cell body, it is present within the central nervous system. It is very interesting that the outflow of the parasympathetic nerves, that is within the brain, from the brain or from the sacral segment of the spinal cord. So it is through the brain, through the cranial nerves 3, 7, 9, 10, or through the sacral segment of spinal cord through segment number S2, S3, and S4. That is why the outflow of the parasympathetic nervous system is called cranio due to this, sacral due to that outflow, craniosacral outflow. And all the cell bodies presynaptic ganglions are starting from the T1 segment all the way to the L2 segment of the spinal cord. So this is the outflow of the sympathetic. That is why it is called thoracolumbar outflow. So this is the outflow of the autonomic nervous system. Let us start about sympathetic nervous system. They originated from spinal cord In the spinal cord, the gray column, you saw ventral horn and saw dorsal horn. In these regions only, where the sympathetic nervous system or parasympathetic nervous system is exiting, there is a special lateral horn between the two. Like this is the dorsal horn of the spinal cord. Here is the lateral horn. This is the ventral horn. Here is the lateral horn. Now this is the what we are talking about. Now this is the white column or white matter of the spinal cord. So this is the part where sympathetic nervous system preganglionic cell fiber is present. From here, cell body is there. The axon exits through the ventral root. You remember the ventral root was the efferent root. The sympathetic outflow, even if it is going to the visceral system, it is still an outflow. So it exits through the ventral root and it carries on pathway, as we will see. It is also called thoracolumbar because it is originating from T1 to L2 segment. That is why the preganglionic presynaptic neurons have spinal cord. They exits through the anterior root, and then there are these words. They connect to the paravertebral ganglia via white remicommunicantes. I will just explain in the next slide. Let us go there. In the spinal cord, here there is a vertebral column. Within the vertebral column, the spinal cord is running. It is containing all the segments and even presynaptic sympathetic cell bodies. Outside the spinal cord, there is chain of sympathetic cell bodies and their fibers. So this is called sympathetic chain. The sympathetic chain contains these dilatations at the regular intervals. These are called sympathetic ganglia. The sympathetic ganglia, their number is as follows. In the neck region, there are 3. In the thorax region, there is roughly 12. We say 11 when the third one of the neck region fuses with the first one of the thoracic region. There are 5 in the lumbar region. Again, it may fuse with the thoracic one to form 4. There are 5 in the sacral region, and there is 1 in the coccygeal region called ganglion impar. Now, now you can see this sympathetic chain that lies outside, it is going much higher and much lower than the outflow tract because the outflow was limited to the T1 to L2 system. Let's see how this whole system works. So this is the simplest, most basic arrangement that preganglionic, let me label as we move along. This is called preganglionic. This preganglionic cell body is present in the lateral horn. This is the dorsal horn. This is the ventral horn. This is the lateral horn. So the preganglionic fibers, they are present in the lateral horn of T1 to L2. From here, this axon, it moves to the anterior aspect, exits the spinal cord through the anterior root, joins the dorsal root to, in the mixed nerves, and as it moves to the spinal nerve, it does not go beyond this point. It has to synapse, and the synapsing cell body is present in the sympathetic chain that we discussed. So there is a connection of spinal nerve to the sympathetic chain, and through that the preganglionic presynaptic myelinated cell fiber, it enters the sympathetic chain. As you remember, it was myelinated, so it was white. So this connection from spinal nerve to sympathetic chain is called white ramus, and in the books you will mostly read white ramus communicantes. Please forgive my spellings and writing. So white ramus communicantes. Within this ganglion, this sympathetic ganglion, there is a cell body. Now this cell body, this is the postsynaptic. This is the postganglionic cell body. Now this cell body, after synapsing, the second cell body, it gives rise to the axon. Now the axon has to be delivered back to the spinal nerve. Now to deliver it back to the spinal nerve, it makes a separate connection than the incoming connection. This connection is called gray ramus communicantes because the second order neuron, the postsynaptic, the postganglionic neuron is unmyelinated. This is called gray ramus communicantes, and it goes and run in the spinal cord, goes to all its branches and supplied the blood vessel for constriction, supplies the sweat glands for sweating, supplies the erector pili muscles to straighten your hair. So this is gray ramus communicantis. So this is the basic circuit of the autonomic nervous system, the basic circuit, I'm sorry, of the sympathetic outflow. But in actual, the story is a little more complex. There are other levels. Why the story needs to be complex? Because this story is very simple. As we it starts from T3, goes to the third uh, thoracic ganglion and moves to the third thoracic spinal nerve. This story is very simple. But as you can see, the sympathetic chain above T1 has also to be supplied. The sympathetic chain below L2 has also to be supplied because the spinal nerves S1, S2, S3, S4, coccygeal nerves, they also need a postsynaptic sympathetic component. So to supply these, this is now we go to the next segment, that is after the for T1 to L2, the emerging from the lateral horn through the anterior root, coming into the anterior root, spinal nerve, and entering the spinal sympathetic chain through white ramus communicantis, that is common for everyone. After reaching the sympathetic chain, there are different fates. The one I discussed already, that it enters through white ramus. At that level, it synapses with the postganglionic postsynaptic cell body, and it exits through the gray ramus to the spinal nerve back. The second option is it enters the sympathetic chain, go up the sympathetic chain or go down the sympathetic chain, then synapse to the postganglionic cell body there and then exit. I will show you the picture immediately after that. And the third option is it enters the sympathetic chain through all the route that we mentioned previously, and it does not synapse, and without synapsing it exits as white ramus, it exits as unmyelinated fiber. So that is the third. Let me show you by picture. This is the case number one that we saw in the picture. It started from the lateral horn. Preganglionic axon started from the ventral root, reached the mixed spinal nerve, entered the sympathetic chain through the white ramus communicantis, synapsed with the postganglionic postsynaptic cell body, and the axon of the postganglionic cell body exited the sympathetic chain into the spinal nerve through gray ramus communicantis, and it went on to supply the different region. And it can go on to supply through the anterior ramus, or it can go on to supply through the dorsal ramus also. This is simple enough. This is a little complex one. For example, to supply all the nerves from above T1, that is the nerve from C1 to C8. Now follow the route. It present in the lateral horn. Follow it here. It's present in the lateral horn, exits through the ventral root, reaches the mixed spinal nerve, gets entry into the sympathetic chain through white ramus communicantis, but it is not synapsing here. Without synapsing, this presynaptic axon, it runs up until it reaches its destination ganglion. It may be first cervical, second cervical, third cervical ganglion that are known as superior, middle, inferior. And let's say it reaches the superior cervical ganglion. Here it synapses with the postsynaptic ganglion, and after synapsing with the postsynaptic ganglion, it exits into the cervical nerve through gray ramus communicantis. And here it's a very interesting difference you can see before your eyes, that here there were white and gray ramus communicantes giving connection to the incoming and outgoing fibers. But as the outflow is only from T1 to L2, the cervical segments will not have any incoming fiber. The no white ramus communicantes, only gray ramus communicantes. We can revise it on the other picture. The similar is true for the fibers going downwards, down below the L2 segment. So in the lateral horn, the cells are present, moving through ventral root, mixed spinal nerve, entering the sympathetic chain through Vitrimus communicantis, then moving down the sympathetic chain until it reaches the ganglion through which it wants to exit. There it synapses with the cell body of postsynaptic neuron, and its axon exits through to the spinal nerve through gray ramus communicantis, and through the ventral or dorsal ramus it reaches the area of its effect. Let's skip this, let's focus on this. This is the third category. In this category, very interesting thing is happening. The synapse is not having inside the sympathetic chain. There are other ganglions present outside in front of the aorta in the abdomen, like celiac ganglion, superior mesenteric ganglion, inferior mesenteric ganglion. The synapses is occurring there. So let's take our signal to that point. So the lateral horn, there is cell body, preganglionic sympathetic cell body is present. It exits through the ventral root, reaches the spinal nerve, enters the sympathetic chain. It still do enter the sympathetic chain through the vitrimus communicantis, but from the sympathetic chain, it does not go back to the spinal nerve either at its own level or by changing other level. It exits directly as its own nerve because it has not synapsed yet. It is still preganglionic, presynaptic myelinated axon fibers. So this is called splanchnic nerve, and it moves all the way through the thorax, through the diaphragm, through the abdomen until it reaches the ganglion that is present in front of the aorta, that is called prevertebral plexus, because aorta is present in front of the vertebral column, and in front of the aorta is present these ganglion. And here it synapses with the postsynaptic axon, and here the postsynaptic fibers, they run along the blood vessels supplying the stomach, small intestine, large intestine, and they supply the sympathetic fibers to them. So these are called splanchnic nerves. So such splanchnic nerves arising from segment T5 to T9, they join to form one nerve that reaches as a single piece to the celiac ganglion, that is called greater splanchnic nerve. The one exiting from T10 and 11 is called lesser splanchnic nerve. The one exiting from T12 is called least splanchnic nerve. The one exiting from lumbar segments L1 and L2, those are called lumbar splanchnic nerves. So these are the three fates of the nerves that can be encountered. You can pause this slide here. This is a picture from Keith Elmore's clinical oriented anatomy. You can go through these. All the fates have been discussed and shown here, but as opposed to the last slide, the presynaptic and postsynaptic have different drawn, like presynaptic are drawn in dotted line and postsynaptic are drawn in solid. You can go through this slide by pausing the video. The fibers, the sensory fibers from the viscera, they also travel back along the same nerve to reach the spinal cord. For example, here the arrangement, the simpler, the first arrangement of the autonomic nervous system is shown like we discussed. Lateral horn, ventral root, spinal nerve, white ramus. Now gray ramus then reaches through gray ramus or effector organ, or splanchnic nerve, excuse me, splanchnic nerve, prevertebral ganglion, postsynaptic nerve organ. So this is the motor component. The sensory component travels along the same nerves back all the way through the sympathetic chain, through the white ramus communicantis into the spinal nerve, and it then goes to the dorsal root horn, and it enters the spinal nerve, spinal cord there. So the afferent fibers, they trace back along the same pathway from which the general efferent fibers came to reach the organs, and they have their cell bodies in the dorsal root ganglions with a pseudo-unipolar neurons. So this was all about the sympathetic nervous system. And as you we talked about the presynaptic, the postsynaptic, where the postsynaptic cells were present, the presynaptic were myelinated, the postsynaptic were unmyelinated, and the presynaptic ones axons were smaller because of the proximity of the sympathetic chain to the spinal cord, and the postsynaptic were longer because of the distance, higher distance, relatively high distance of the sympathetic chain from the effector organs. So let's talk about the parasympathetic nervous system. Their outflow is craniosacral. They originated from brain in the form of cranial nerves. There are four cranial nerves which contain the parasympathetic components. You must memorize them. Cranial nerve number 3, 7, 9, 10. And from the spinal segment, my apology for this, it is not S1, 2, 3. Yes, it is S2, 3, 4 is the better answer, but variation do occur. So these fibers that are originating from the cranial nerves, that are originating from S2, 3, 4, those are the presynaptic fibers. They have to uh, synapse in a ganglion before uh, going to the effector organ. So in this case, there is no well-defined chain as we saw in the sympathetic system. They have their own ganglion depending upon the regions. For the nerves exiting from the head and neck region, there are four very prominent ganglions, like the cranial nerve number 3, the oculomotor nerve. It contains the presynaptic parasympathetic fiber that supply the area around the orbit, the muscles and eyes, and around the orbit. So there is a ganglion called ciliary ganglion. The ciliary ganglion is made up of cell bodies of postsynaptic cells that will ultimately go to the eye. Similarly, cranial nerve number 7, it goes to two different ganglia. Presynaptic fiber may go to the pterygopalatine ganglion. After synapsing, they go to and supply the area of the nose and palate. Or it may go to the submandibular ganglion, the postsynaptic cells that are present in the mouth region that go in to supply the glands of salivary glands. And then there is otic ganglion that is receiving presynaptic from glossopharyngeal, and it is going on to supply the parotid region, this parotid gland. Apart from these four ganglions, for the other organs which are receiving parasympathetic supply in the heart region, in the thorax region, they have their respective plexus. There is cardiac plexus that contains the cell bodies of the presynaptic, postsynaptic cell bodies. There will be pulmonary plexus. There will be esophageal plexus. Similarly, there will be enteric plexus in the intestine, small intestine. Like there will be. So the interesting thing about this is the postsynaptic cell bodies below the head and neck region for the parasympathetic nervous system, they are embedded within the walls of the organs. Like in the thorax region, the plexi are present attached to those organs. The cardiac plexus is present here. In the case of intestine and genital and urinary system, it goes a step further. The plexus is actually embedded within the wall of those organs. So the postsynaptic cell bodies are actually present inside the walls of those organs, and very short axons have to move from uh, this postsynaptic cell body to the affecting part. So they have a very long presynaptic component and very short postsynaptic component. So this is how that is shown here. Like cranial nerves at the exit from the brainstem, they reach through the cranial nerve to the ganglion. This is that point of synapse. Before it was presynaptic, after that was postsynaptic, and it reaches the organ. In case of small intestine, large intestine, the cranial nerve reaches inside the walls of the viscera. There is the cell body of the postsynaptic that supplies the affecting glands or the muscles. The same hold true for the exiting the spinal segments, and they have initial segment like same. They will be present in lateral horn and they will exit through the ventral root until they reaches the viscera, and within the viscera they will have postsynaptic cell body. So that is about that. Peripheral ganglia are usually close to the viscera. In the cranial region, there are ciliary, pterygopalatine, submandibular, and otic ganglion. In the sacral region, they have different plexi like hypogastric plexus and walls of the viscera. Postsynaptic, just like in sympathetic nervous system, are unmyelinated and are very short as compared to sympathetic. And again, the afferent fibers that travel back in the same direction to reach the sensory ganglia in either the cranial nerves or the dorsal root ganglions in the spinal nerve. So here is a very nice picture, one of my favorite ones from the Snell's Clinical Anatomy by Regions, that shows all of those pictures. It has shown parasympathetic in blue, sympathetic in red, preganglionic in solid lines, and postganglionic in dotted lines. You can discuss and go through this picture. This has almost all of the theory that I discussed in the last 20 to 30 minutes in this one picture. You can ask in the comments below if any confusion remains. The in the small intestine, in the digestive system, the nerve plexus that are reaching, they are very well organized into two plexus at two levels, in the submucosal layer and between the muscle, that is myenteric layer. So there is submucosal plexus and there is myenteric plexus. This is a very extensive system containing about 100 neurons, 100, I mean, 100 million neurons. There are motor neurons, there are sensory neurons, there are interneurons, and contains lots and lots of postsynaptic parasympathetic neurons. So they get signals from mostly the input signals from the presynaptic parasympathetic and postsynaptic sympathetic, and they influence the system. However, this system is so extensive that even if it is cut off from all the incoming signals, it still work. If the intestine is isolated from all the nervous system of the body, the peristalsis will still work. So it is the mini brain within itself. So in humans, the inputs from the sympathetic and parasympathetic system does not actually go to run it, they go to influence it, they modulate its already running functions. So that is why it's an third division of nervous system also. It's also called an independent division of nervous system, the enteric nervous system. So that is the one that is present in submucosa, that is submucosal plexus, that is present in between muscle layers, that is myenteric plexus. So that's it. I hope so you got something from this lecture. I know it was a big one. I will be marking all the chapters below that you can jump on and listen to the part you want to listen to. If you have any question regarding the content of this topic, if you have any suggestions for me or feedback for this session, please do mention in the comments. I will try to get back to you. And if any improvements are to be needed, I will try to improve myself in the upcoming session. Thank you so much for listening. Stay tuned. Till next time. Goodbye.